So we're looking at the four key biological macromolecules. So macromolecules, just it's large molecules. Carbohydrates are probably the first one that people think of when you think of these, and that is the complex name for sugars and their polymers. The simplest carbohydrates are going to be the monosaccharides or simple sugars, something like glucose, which you can see here. Those come together to form disaccharides, or the double sugars. That's going to be something like lactose. Okay, so if you're lactose intolerant, for example. Polysaccharides are going to be when you have polymers of many monosaccharides. The key idea here, though, is that carbohydrates are macromolecules. They are also polymers. So they're going to be made of repeating subunits that are the same. So glucose is the example of a monomer of carbohydrates. It's not the only one. There are other ones like pentoses and trioses, but glucose is a 6-carbon monomer, hexose, and it is one of the most common. Whenever you put two sugars together, that's going to be dehydration synthesis. That is in sugars, it's called a glycosidic linkage. So we get the base idea. Polysaccharides are significantly larger. It could be dozens to hundreds to thousands of these repeating subunits forming the polymer called a polysaccharide. The polysaccharide, you are expected to know 5 polysaccharides. The base 3 are probably the most important for you, though. If you'll notice, if you look at all 3 of these, starch, cellulose, and glycogen, all 3 of these are the same thing over and over and over and over. It's glucose found in all sorts of structures. Now, the thing is that there's different types of glucose. You have alpha glucose and you have beta glucose. These are isomers, and they're going to alter that structure. Starch is going to be a storage polysaccharide for plants that are composed entirely of glucose molecules. But starch is going to be made of alpha glucose. It's going to all be bonded in the same direction, which makes it very easy to digest. Cellulose, which we also call fiber, is going to be made of beta glucose in alternating directions, which you can see down on the bottom right. That is going to be much harder to digest, and it's why we can't digest fiber. So cellulose is a structural polysaccharide in plants. Starch is a storage polysaccharide in plants. Glycogen is the animal version of starch. So glycogen, if you look above, glycogen is going to be a very branched storage polysaccharide. We know it's storage because all of those bonds are in the same direction. They're all alpha. Okay. Now there are different types of things, like starch, for example, has 2 major types, amylose and amylopectin. So they're going to be different sort of fixture structures, but they're going to be bonded in the same way. So starch, cellulose, and glycogen are all glucose bonded differently and structured differently. There are other polysaccharides, for example, chitin and peptidoglycan. These are polysaccharides that you're expected to know because they are part of cell wall polysaccharides. Plants use cellulose in their cell wall. Fungus and insects use chitin in their cell walls, and then bacteria use peptidoglycan in their cell walls. You'll notice that these guys, however, are different because they have nitrogen. Okay, so just because it's a sugar doesn't mean that it can't have other things. It's just not one of the common structures. Okay, but you are expected to know the 3 cell wall polysaccharides and the differences between them. You do not have to recognize it from a picture, but if you see a complex sugar like this and you see nitrogen, you know it's one of these 2. Okay, and as well as the 3 glucose ones that we looked at on the previous slide. Lipids are a little bit different. Lipids are composed of different subcomponents. There's not one monomer. So lipids are macromolecules, but they are not polymers. Okay, so lipids, there is no sort of that unifying nature. The thing that unifies lipids is that they are non-polar covalent. That means that they're not going to dissolve in water. The most basic kind of lipid is a fat. It's a triacylglycerol. That's going to be made of 3 fatty acids attached to a glycerol. One of the ones that you might be more familiar with is the phospholipid. You can see the picture over here. So that's 2 fatty acids, a glycerol, and a phosphate group, phospholipids. Okay, it's not the same thing over and over and over again. They are different. Okay, lipids are also going to include things like waxes and steroids, like cholesterol. Okay, so again, the unifying thing here for lipids is that they're nonpolar. Okay. Now, when you deal with fats, fats then can be sort of subdivided into saturated and unsaturated fats. Saturated fats are going to be in a straight chain because all of the carbon-hydrogen bonds are single. There's nothing to cause a bend or a geometric change. However, if you alter that, if you make it unsaturated, there will be double bonds. And when you start looking at that on a molecular level, every time there is a double bond, it introduces a bend or a kink in the chain. The more bends in the chain, the more unsaturated it is, the more space there is between the molecules, the more liquid it is. So the more saturated fats are going to be solid at room temperature, whereas the more unsaturated fats, or even polyunsaturated fats, are going to be liquid at room temperature. Like vegetable oil is a unsaturated fat, whereas butter is a saturated fat. Now, what about things like margarine? Okay, margarine is something where we introduce another idea, which is called hydrogenation. If we force extra hydrogen into the molecule to force all of those double bonds to break and become single bonds, you create a fake saturated fats. You're able to turn vegetable oil into a solid, and we use margarine. Why? Because the margarine is healthier for you biologically. It doesn't stick to your blood. It's not going to contribute to like heart disease and that kind of thing. So a diet that's rich in saturated fats contributes to plaque deposits and heart disease, whereas the unsaturated fats typically don't. Like omega-3 fatty acids are essential fatty acids. We need them, but we need to find some ways to bring those into our diet. Proteins are macromolecules. Proteins are polymers. Okay, they are polymers. That means that the monomer is something we need to be aware of. The monomer of a protein is what we see here. It's an amino acid. Within the amino acid, you have an amino group on the left, a central carbon in the middle, and then a carboxyl group on the right. That means that you have a base on one side, an acid on the other. It is both an acid and a base, which means it's a buffer. It can go up or down depending on what it needs to do. Every amino acid will be made of the same components again and again and again. Putting them together, dehydration synthesis, is called a peptide bond, and that is going to create this chain of proteins that you're used to seeing. Now, what is different in the monomers is the so-called R group, or the variable group. So the R group is the only thing that's different. Looking at the protein, NCC, 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 it's the same thing over and over and over again. What's different is the side chains on the outside. Okay. Remember, sulfur is one thing that I can use to tell proteins because sulfur is involved in two amino acids, the methionine, which you can see here, and cysteine, which you can see here. Okay, so that sulfur is very useful. Why do we care about proteins? Well, proteins do a lot in biology. Proteins can be enzymes, okay, which means that they're going to aid as biological catalysts. Hormones. Okay, you have protein-based hormones. They're going to be used for cell signaling and communication. You have storage proteins like albumin. Okay, you have motor proteins making up your muscles. You have defensive proteins like antibodies. You have transport proteins moving things across cell membranes. You have receptor proteins using for signaling between cells. Okay, and you have structural proteins like collagen, keeping your skin all flexible. When that begins to break down, you start to wrinkle. Okay. The function of protein depends upon its ability to sort of recognize and bind, and that is going to be something that we deal with a lot in the next unit. But proteins are very flexible in their function. Another thing that separates proteins from carbohydrates and lipids is that it has both directionality and levels of structure. There is a beginning and an end, a left to the right, which isn't something that we saw in carbohydrates or lipids. Okay, so proteins, the directionality is determined by the NCC relationship. Remember, the carboxyl group has the OH group on the right, so we will only be able to add new amino acids on the carbon end. So there's going to be the amino acid end and the carbon end. We can only add new ones to the carbon end. So that gives me directionality. The directionality is from N to C. We also have levels of structure. There are 4 levels of structure to a protein, and you must know these. The first one is just the straight covalent bonds holding this protein together. Okay, so that's just the sequence of the amino acids moving across this whole primary structure. Okay. The secondary level of structure is going to be when the NCC backbone interacts with each other. Okay, the nitrogen carbon, the R group has nothing to do with the second level of structure. It's just the NCC. So all we can really have here is hydrogen bonds. Do they bond across or next? That's really the only difference. So there's really two possible structures at the second level, which is convenient. The alpha helix, which is a coil held together by hydrogen bonding between every fourth amino acid. Okay, and then you have the beta pleated sheet, when you have two or more regions of the chain lying side by side connected together. Okay, pleated sheets are found all sorts of the place, lots of things, and the presence of all those hydrogen bonds makes it incredibly strong. Okay, so like spider webbing, for example, has a lot of beta pleated sheets, which is why spider silk is so strong. Okay, the third level of structure, the tertiary structure, that is going to be much more complicated. Why is it so complicated? The tertiary structure is complicated because this is where the R groups are involved. Because the R groups are so different from protein to protein to protein, there's going to be four major ways that they can interact. They can form hydrogen bonds, okay, fairly self-explanatory. Okay, they can form ionic bonds because this is forming inside of the protein and away from water. Ionic bonds are possible. Okay, hydrogen bonds are partial charges, ionic bonds are full charges, but they're essentially working in similar ways. We can have hydrophobic interactions. That means that we have non-polar R groups folding away from water and polar R groups folding towards water. Okay, these ones are all carbon and hydrogen, very much a non-polar lipid-like structure. They're going to be on the inside, whereas polar ones are going to be on the outside. The fourth thing that can separate the third level of structure is the disulfide bridges. So if we have two cysteine amino acids that are parallel to each other, they can form a disulfide bridge, a strong covalent bond that is going to act like a rivet holding these proteins together. Okay, lots of proteins stop at the tertiary level of structure. Some proteins will also have a fourth or quaternary level of structure. That is when you have an aggregate of two or more tertiaries. Okay, for example, you have hemoglobin on the bottom right here. That's made of four different subunits put together, four different tertiaries, making a single quaternary. Okay, collagen is made of three. Okay, but you get the different idea. So if you have multiple tertiaries coming together, that is a quaternary structure. Why do we care so much about protein shape? Well, without those proper shapes, we have many biological problems. Okay, we actually have structures that help within these things. Chaperonin is a great example of this. Most proteins appear to undergo several intermediate stages before they reach their mature structure, and the folding of these proteins is assisted by chaperone proteins. They don't specify the final structure, but they segregate and protect the polypeptide so that it can fold properly. If proteins misfold, we can end up with many diseases and structures like this one here. We have a protein aggregation of misfolded proteins leading to an amyloid plaque, leading to Alzheimer's. Okay, proteins can be denatured. Their structure can be lost. Okay, denaturing can be due to things like physical force, temperature, salinity, acidity. But if you denature a protein, you will lose its shape. Okay, because it's misshapen, the biological protein is typically biologically inactive. Most proteins become denatured if they're transferred to new environments or altered new conditions. High fever, for example, can be fatal because proteins in the blood become denatured by the high body temperatures. Some proteins can return to their functional shape. They can be renatured, but not all of them. The accumulation of incorrectly folded polypeptides is associated with many diseases like Alzheimer's, Parkinson's, but some of the more scary ones are the so-called prion diseases. Prion diseases are protein molecules that have no nucleic acid genetic information. The proteins are made up of amino acids, and there's some sort of defect that causes them to fold incorrectly into some sort of structure. Mad cow disease, Creutzfeldt-Jacobs, Scrapie, Kuru, these are spongiform prion diseases because the misfolded proteins then go on to misfold other proteins, leading to literally holes, in this case, in your brain, which is what drives you insane. So prions are misfolded proteins that are pathogenic, and that is incredibly terrifying if you really think it through. We do see that even a slight change in the primary structure can affect a protein's conformation and its ability to function. The substitution of one amino acid for the normal one in sickle cell okay, in a red blood cell, going from valine or valine to glutamic acid, that causes the red blood cell, instead of folding outwards in a hydrophilic shape, to fold inwards in a hydrophobic shape, leading to the many negative impacts of sickle cell. So the changes don't need to be extreme. It can be a single subunit altering everything else. So shape and bonding and structure are incredibly important to protein functionality. Our last biological macromolecule that we're looking at is the nucleic acid. Okay, nucleic acid, this is things like DNA and RNA. They're going to be made of nucleotides as the monomer. The nucleotides we put together through dehydration synthesis process called phosphodiester bonding. The nucleotide is made of a phosphate group, a 5-carbon sugar, and a nitrogenous base. Okay, of the common forms of DNA and RNA, you're expected to know that there are 2 types of pentose 5-carbon sugar. You have deoxyribose and ribose. The difference is the hydrogen or the OH group here. Okay, if you really break it down, what you do is you number the carbons. So you have carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. The second carbon determines whether it's deoxyribose or ribose, and that is what separates the D from the R. Okay, another difference is that DNA is typically 2-stranded, where RNA is typically single-stranded, but that is not the be-all, end-all, so I wouldn't use that as the major separator. Okay, within this other structure, we also realize that there are 5 common nitrogenous bases. You have the 3 pyrimidines, okay, C, U, and T. I always remember that you had to cut the blocks of the pyramid. Okay, these are single-ringed nitrogenous bases. And then you have the 2 purines. I always remember this as pure silver, or AG, pure silver. These are double-ringed structures. Okay, so every nucleic acid is going to be made of these 3 things. A phosphate group, which is the same for everything, 5-carbon sugar, one or the other, and then a nitrogenous base, one of the 5. Okay, DNA will use thymine, RNA will use uracil, so that's the 3rd difference that we can use to separate these structures. So polynucleotides are synthesized when adjacent nucleotides are joined by covalent bonding, like we said, in a phosphodiester linkage between the OH groups on the 3rd carbon and the 5 carbon of the next. So DNA and RNA, just like proteins, have directionality. In proteins, it was N to C. Okay, for nucleic acids, it's 5 to 3, so it's kind of rhymey. Okay, the other thing that we see is that the bonding that happens is between A and T and G and C in DNA. In RNA, change T to U. That means you're always going to have a 2-ring structure bonded to a 1-ring structure, meaning that it's always going to be 3 across as far as the nitrogenous bases, keeping it the same distance all the way. And that's something that's important there. The other thing that we realize is that DNA is anti-parallel. If one side is 5 to 3, the other side is 3 to 5. We can see that relationship here. Okay, so DNA is complementary, A to T, G to C. It's anti-parallel, 5 to 3, 3 to 5. It's always 3 nitrogenous rings apart from each other. Those nitrogenous rings are held together by hydrogen bonds. We can see that here. Between G and C, there are 3 hydrogen bonds. Between A and T, or A and U, there are 2. So that's how it knows what to use. And that's sort of the base structure there. So we've looked at all 4 of the major biological macromolecules. 3 of them were polymers. Lipids are not polymers because there is no repeating subunit, but we should have a basic idea now of how macromolecules work.